Anna, and I'm based in Boston area. And today we'll go over some of the topics that might interest you guys when it comes to Ingenuity pathway analysis. So this is just our legal disclaimer. The product that we are showing today is intended for molecular biology applications only and not for diagnosis, prevention, or treatment of a disease. And like Scott mentioned, we have a nice raise hands button and Q&A box at the bottom of your Zoom screen. So along the way, during this training, please feel free to type in your questions into the Q&A box. It helps me to track those questions, and then I can answer either along the way or basically this session is divided into 3 parts. We'll answer the questions after an hour after 1 hour sessions. So that way we address at least a lot of your questions. So in terms of agenda, we are going to divide this session into 3 parts. Since the training is for 3 hours, we'll try to dedicate an hour each for some of these sections. So the first topic we'll go over is how to leverage IPA when you don't have any data set to work with. So let's say you don't have any RNA-seq data, you don't have omics data. How can you still utilize IPA for your research, publish papers using this existing public knowledge? So initially we'll start with this network construction feature where you can generate a hypothesis by simply searching for a gene or a function or a disease that you want to look at, and generate a custom network from scratch and perform in silico prediction. Then we'll also look at how to look at some activity prediction for key regulators, proteins, pathways, or functions that you might be interested in, and see how these entities behave in other public data sets. Now, after the live Q&A, then we'll go over another feature, which is Land Explorer, where if you are working with a biomarker signatures or a gene that you're working in the lab that you found with your Western blot results, you want to see how this gene is expressed in different diseases or different drug treatments or different cell types. So you can easily utilize our Land Explorer feature to look for gene or biomarker expression. Then we'll also look at how to search for public data sets, especially public transcriptomics analyses. So if you are working, let's say, in infectious diseases area or neuroscience area, and you want to search for relevant data sets coming from published studies, you can easily search for them, and you can also look at the interpretation of this data set. And maybe you'll find some new novel insights from these public data sets with which you can then publish a brand new paper. And finally, we'll look at, in the last session, how to process your own data set. So if you are working with right now currently in RNA-seq data, single cell data, proteomics data, or are planning to generate this data in near future, you can still leverage IPA to process your own data sets. And then we'll see how to generate these analyses, how can you compare multiple experimental time points or treatments, and then how to compare your own transcriptomics data with other public data sets. So with that, I just wanted to briefly mention that now when you search for IPA on, let's say, googlescholar.com, you're going to find lots of papers where people have utilized IPA for their research. So what I have done is I have gone through some of the recent studies and pulled up some examples where the researchers leveraged IPA for their research. So this will give you some idea about the type of data can be used coming from different systems or different outputs as well. So one example over here is this researchers looking at hepatocellular carcinoma. So this is a cancer area research, and they found these critical regulators that play a role in the development and progression of this cancer type. So they specifically utilized the Canonical Pathways feature and identified which key pathways were activated or inhibited. And they were able to also look at how this particular pathway was behaving when some of the genes were up or down regulated in their data set. Similarly, so this particular researchers, they looked at microglial microRNAs, especially when it came to tau pathology. So if you are interested in identifying key mRNAs and microRNAs targeting your disease area, you can easily utilize IPA for that. And you can here clearly see key regulators that were either activated or inhibited in their disease pathology, and they were able to identify key microRNAs through Untitled pathway analysis as well. Now, this is a unique paper where the researchers, they utilized IPA and existing public data coming from GEO and SRA. So here they did not use any of their own data. They took these public data sets. So here you can see the list of public data sets that they utilized coming from mouse and human, and looking at especially at liver regeneration. And they were able to generate a novel hypothesis, and they were able to extract novel results from this by analyzing this public data, reanalyzing the data using IPA. So just imagine if you want to publish more papers, or let's say even write a grant, you can still utilize IPA by just taking existing public data sets and reanalyzing them in your own way. And apart from the publications, a lot of our users, they have utilized IPA for grant writing. So in this slide, we are looking at the NIH reporters statistics reporting amount of funding that was provided to researchers who were working with Untitled pathway analysis. So in the past 3-4 years, over $5 million was awarded to researchers who used IPA for their research. And these, as you can see, these are the different institutions that gave funding to people who utilized IPM. So we can see a lot of applications when it comes to IPA. So you can publish papers, write review papers. You can even generate hypotheses just for your grant writing. Now, one of the unique things about IPA is that IPA's database is not only comprehensive, but it also shows you directionality and causality. So when you come in with data such as RNA-seq data or proteomics data, you want to upload this data set into IPA, and you want to utilize the different tools that we have to generate these different visuals, graphs, and networks to identify novel pathways, biomarkers, drug targets, and just discover new disease mechanisms. So let's say a user like you have this set of genes that are either upregulated or downregulated. And with this information, you can imagine it is difficult to interpret which genes are important or how they are involved in a particular pathway. So when you upload this data into IPA, what IPA will do is it's going to compare the directionality that you have in your data with our knowledge base, which contains over 8 million data points that we have extracted from the literature. And then IPA will tell you if the pathway was enriched in your data set and whether this pathway was activated or inhibited. So here in this bar chart, you can clearly see that we have a list of pathways that were enriched in a data set that we were working with. And you can see that we have different colors on them. So here you can see that NRF-mediated oxidative stress is enriched and it is predicted to be activated just by looking at this orange colored bar. And this particular pathway is shown in blue color. It means that this particular pathway was inhibited based on the expression pattern you had in your data set. Now here in this particular bar chart, we can see that, sorry, in this particular network diagram, you can see that our FN1 gene here is shown in green color, which means that it is down-regulated in your data set. So it had a negative fold change value. And based on IPA's prediction, you can see that cell motility and cell adhesion, they're also reduced when FN1 gene was down. So just by looking at the pathways, the shapes, the colors, the connections in the network, you can generate a strong testable hypothesis. Now, in case if you want to compare different time points, treatments, etc., in IP, you have a nice feature where you can generate these node charts. So that will display different experimental groups in one single plot. So here you can see that TRXR1 gene is differentially expressed in these different conditions. So it is up in condition 1 and it is down in condition 2. And in case you're working with multi-omics data, you can see that in condition 1, not only the mRNA expression goes up, you can also see that protein expression went up in this particular condition. So you can essentially compare your time points, treatments, different experimental conditions, and you can also compare multi-omics data. You can compare your proteomics with RNA-seq, etc. Now, apart from the plots and networks that I just showed you, you can also compare multiple data sets side by side by generating heat maps. So these heat maps are typically found, let's say, in a lot of published papers, especially those who perform some sort of RNA sequencing, single cell sequencing, etc. So if you have any type of omic data, you can compare them side by side and in form of a heat map. So here you can see we have RNA-seq data and we are comparing it with proteomics data. And you can also see we have 3 different time points that you're working with. So this is day 4, day 9, day 23. So these are different time course treatments that was performed for these omic data sets. And as you can see, this fatty acid beta oxidation pathway is activated in a time-dependent manner in both RNA sequencing data and in proteomics data. And that's because you can see there is this shade of orange color. It increases over time. So you can see that at day 4, the shade of orange color is slightly dimmer, and you can see you have a intense orange color by day 23. So this tells you that there is a time-dependent activation of fatty acid beta oxidation in this particular data set. Now, apart from comparing your own data, you can also compare your data with any of the public data sets side by side as well. So let's say you have some sort of RNA sequencing data that you generated in the lab, and you also wanted to see if there were other papers or other researchers who did similar analysis, or maybe they used a drug, utilized a drug to treat the similar disease condition. So you can find those public analysis and compare them side by side with your own data set as well. And then you can determine the biology behind what's going on. And all of this that I just showed you is possible because IPA is driven by its powerful knowledge base, and it has over 8 million literature-based data points that we have curated over several decades. And this allows IPA to interpret any type of omic data, and you will be able to generate all these results from IPA. Now, like I mentioned when we were discussing the agenda, you can utilize IPA in 2 different ways. One way is you leverage IPA when you come in with some sort of data like RNA sequencing data, proteomics data, metabolomics data, and then you can extract biological insights from that data. And another way is you can come in with IPA without any data, and then you can ask a question like, how does my target gene mediate a disease downstream? What are the upstream regulators of my target gene that you're working with? So this will allow users like you to generate a novel hypothesis, and you can also test this in silico. So let's say how we can utilize IPA when you don't have any data set to work with. So let's say you don't have any transcriptomics and proteomics data, but you're still interested in mining IPA's knowledge base and generate a new hypothesis. So let's take a scenario. So let's say you're working with a target gene or a protein that you're working in the lab, like PDCD1, and you want to see what happens when you block PDCD1 or you downregulate PDCD1 in silico, and what happens to activation of T lymphocytes. So you're working with T cells in the lab and you want to see what will happen if I block this protein. So you can easily search for your favorite gene and the function that you're interested in IPA, and you can easily generate a network connecting your target gene to this function that you're interested in. Now, in addition to that, IPA will also show you directionality and causality. So let's say you're interested in using a drug and or using CRISPR and you want to downregulate this gene, and you want to see the effect on this pathway and this downstream biological function. So you can easily label this with green color and say that I want to downregulate this gene. And you can generate this prediction in silico as well. So here you can see that when PDCD1 is down or inhibited, or using CRISPR, we have inhibited the expression of this gene, you can see that IL-2 or TNF, they are activated. IL-13 and STAT1 are inhibited based on orange and this blue color that you see here. And it ultimately leads to increased activation of T lymphocytes. So this is how you can easily search for your gene and function and then generate this hypothesis in silico. And on top of this a generation of hypothesis, you can also detect if there are any drugs that you can utilize to target this pathway. So you can see that this pembrolizumab is a drug that you can target with PDCD1 in the lab, and you can test this hypothesis. And you can also read more information about the genes that are part of this network. You can read how some of these drugs were utilized in past by looking at the clinical trial data. And you can also see how each gene or node is connected to each other, where this information is coming from, why this prediction was made. So you can look at the relationships between two nodes by looking at our findings information, which is to see how these nodes were connected to each other. So let's see how we can generate this network within IPA using our knowledge base. So with the raise of hands, how many of you would be interested in generating this network, the one that I just showed you? All right. So if you have any questions regarding this network generation, so please feel free to type into the chat box, or sorry, the Q&A box, and then we can address the questions along the way as well. So when you open IPA on your computer, this is what the lay of the land looks like. So in the center, you have the quick start menu. So this gives you a brief information about what are the different ways you can utilize IPA. And it also gives you information about if there is a new release or new features that are going to be added within IPA. Another thing I like to point out is our contact support button here. So if you have any questions ever regarding your analysis or how to run some of the features, how to interpret the data, you can always reach out to our support here on this email, tsbioinformatics@qiagen.com. All right, and then here for the network construction, we are going to utilize this section on top, which is our search bar. So here, first step we are going to do is we are going to go under genes and chemicals and type in our favorite gene. So here I'm going to type in PDCD1. So if you have IPA open on your computer, you can search for your favorite gene, or you can just follow this along with me if you're interested. So we type in PDCD1, make sure this is under genes and chemicals, and click search. And here you can see we have our search word that we were interested in. And you can see the synonyms for this gene, where it is located, if it was utilized as a biomarker, or what drugs have been identified to target this particular protein. So the first step we'll do is we are going to select our gene that we are interested in by checking this box. And then we are going to click on Add to My Pathway. And here we are going to click on New My Pathway because we want to create a brand new pathway. And this is going to open up a blank page for us. And here you can see we have PDCD1, and you can click on it and you can move it around. Now let's go back to our, I'm going to close this search box here, and let's go back to our top panel. And under diseases and functions, we can write activation of T lymphocytes. So here you can see while I type in the keywords, some relevant terms will show up, and I can see activation of T lymphocytes showed up, and I can select that option. And this is going to open up in the search page. And just like we did with the gene, you can select the function. So you can search for a disease if you want. You can search for cell migration, or some any keywords that you're working with, and just search the relevant terms you want. So we select the function that we are interested in, and we go back to Add to My Pathway. And then instead of creating a new pathway, you want to add it to the pathway that you just created. So here you can see I have pathway number 1, and here we see an option for pathway 1. So we select that. Now the pop-up here asks you, do you want to add the molecules that are associated with activation of T lymphocytes? So you can see there are 815 molecules associated with this. Or you can ask, or do you want to add the function as a node? So here we are going to use the middle option because we want to add this node as a function. And we can click OK. And here we have PDCD1 on one side and activation of T lymphocytes on the other side. So you can move them around. Now from here, what we are going to do is we are going to construct a network connecting PDCD1 to activation of T lymphocytes. So to do that, we are going to use this build button. And under build, you're going to use this drop-down menu, and we are going to utilize this path explorer. Just like the feature term says path explorer, we want to find the path connecting our PDCD1 to T lymphocytes. So here, first thing we are going to do is we are going to click on PDCD1. So you see when you click on a node, it is highlighted in blue color. And then we are going to add this to set A. So here we clicked on it and we say add to set A. Then we are going to do the same thing with our activation of T lymphocytes. And then we are going to add this to set B. So here we are going to add this to set B. And then we are going to set a direction. Now, imagine when you're working with cell biology. So when you think about it, you want to block or inhibit this receptor or transcription transmembrane receptor, and then you want to see what happens to the function downstream. So in that scenario, you're going to set the direction from A to B. You want to find relationship. You want to find key molecules that are connecting PDCD1 towards activation of T lymphocytes. And the final step here is we are going to click apply. So we added PDCD1 to set A, then we added PDCD1, the activation of T lymphocytes to set B, then we set the direction, and then we are going to click apply. So once we do this, IPA is going to search for all relevant molecules that are connecting PDCD1 to activation of T lymphocytes. So here you can see at the bottom, we have a table that shows a connection. So there is an existing connection directly between PDCD1 to activation of T lymphocytes. So we can check the box next to them and we can add them to our pathway. So you can see this is connecting the two entities. Now, if you want to find additional genes, proteins, or molecules that are connecting, we are going to go under the shortest path +1. So here you can see when I click on the drop-down menu, you see this path +1 in parentheses, we have 48. So it says that there are 48 other paths connecting PDCD1 to our function. So here you can see that PDCD1 is connected via CD274 to T lymphocytes, similarly via IFNG. And if we scroll down, you're going to find the rest of the 48 connections that we have. So we are going to check all of these, and then we are going to add all 48 connections to the pathway. So there is a question in the chat box, in the question Q&A box. It says, which species in this analysis in, and is this limited to mouse or human? So this, the analysis is including every relation coming from both mouse and humans. So if you do wish to find relationships that are limited to specific species, you can go under the species option filter here. And if you click on it, you're going to find some options for species. So let's say you only want to find human literature-based evidence, only evidence coming from human studies or mouse studies or rat studies. So you can select and filter the options like that. So filter for this, and then you can say, click apply. Similarly, if you were to look for, okay, I only want some protein-protein interactions or some sort of causation relationship. So you can go under relationship types and say, I only want some sort of phosphorylation-based relationship. I am looking for only protein-protein interactions. So you can filter for those type of connections as well. So here what we have is a network that looks pretty similar to what we had on my slide deck. And from here, we can see that PDCD1 is connected via these 48 molecules towards T lymphocytes. Now from here, we want to, our goal was to test this hypothesis in silico. What will happen if we block or inhibit PDCD1 using, let's say, a monoclonal antibody, or you want to use a CRISPR on this? So to test this hypothesis in silico, we are going to go under this overlay button. And under overlay, we are going to click on the drop-down menu. And here we are going to go under this option called map feature, which is molecule activity predictor. So what we have now is these two different colored buckets. So within IPA, red and green colors have a meaning associated with them. So whenever you have red color, it means you are increasing the expression, increasing the measurement, you are overexpressing a gene. Whenever you use the green colored bucket, you are trying to decrease the expression or block the expression. So that's why it says decreased measurement. So here in this scenario, we want to block or reduce the expression. So we are going to choose the green colored bucket. And then we are going to click on the node on PDCD1. And as soon as we do this, you can see the prediction shows up coming from IPA. So here, when PDCD1 is down, it leads to inhibition of STAT1, right? So STAT1 is inhibited. You have IL-12A that is inhibited. And you can see that IL-2, TGF-beta-1, they are activated. So whenever you see orange and blue colors, as you can see in this legend, these are predictions made by IPA. So orange is activation, blue is predicted inhibition. And this ultimately leads to activation of T lymphocytes. Now, you might be wondering where this information is coming from. How did IPA make these predictions? So to find that information, what you can do is you can simply double click on these connections. So if I click on this connection between PDCD1 and IL-2 and double click on it, this is going to open up this relationship summary page for you. And this is going to tell you why IPA made this prediction. So this prediction was coming from at least 8 different papers based on expression relationship. And to locate those papers, you are going to click on this hyperlink. And this is going to open up a web page for us. And this is going to give you the list of all the papers that were talking about relationship between PDCD1 and IL-2. And if you want to read more about the paper, you can go here, click on the hyperlink, and this is going to open up the paper for you itself. So these are all the 8 relationships or 8 papers that from which we made this association how PDCD1 is connected to IL-2. And you can do this in the same way you can do this with any other connections. You can click on any lines that are connecting 2 nodes with each other. So if I do PTPN6 and do this with activation of T lymphocytes, this is going to give you papers talking about that. So how this gene was knocked out in mouse T cells, and this increased the percentage of activated mouse CD4 positive T cells. So this is how we made the connection and how this hypothesis was generated. So with the raise of hands, how many of you would find this useful for your research? So I see a lot of hands are being raised. Thank you guys for doing that. Now from here, you can do a lot of interesting things. So if you want to find any drugs that can be utilized to target this network, you can easily do that by going under overlay. And here you can see we have an option for drug. And if you click on it, you can see these are all the drugs that are listed that can target the genes within this network. So let's say if you want to find something that targets PDCD1, so I can sort this by alphabetically. If you hit on the header for targets, and then you can maybe find all the drugs that are only linked to PDCD1. So here you can see we have nivolumab that shows up. And once you click on this, you can see the nivolumab is a drug that can target PDCD1, and you can add this on top of your pathway. And similarly, if you want to find if there are any biomarkers that have been utilized in past within my network, you can go under overlay again, see this. So we have a biomarker option here. And you're going to find the list of biomarkers for all the known diseases out there. So here, so for example, so these are some of the genes that were utilized. Let's say for ovarian cancer, at least 11 genes from this network was utilized for ovarian cancer efficacy. So it was these were used as biomarkers. So you can easily find the right relevant information, or for example, here you can see some of the markers were utilized for Parkinson's disease. So that's a unique new information. So you can, if you're working in neuroscience area or neurological diseases, so this is a very unique information that you can identify. So you can add this as a tag. So, so from here, you can also easily, nicely arrange this network. So let's say now this network is ready, the predictions are ready, and you have all the relevant information that you want. But then you want to publish this, or you want to add this to a grant in a form of a very nice figure. So to do that, you can go under this change layout button. And here you'll see we have lots of options. So you want to do organic view, circular view, radial view. So one of the popular ones among a lot of our users is this subcellular view. And here you can see that all the genes within your network are now arranged based on their location within the cell. So you'll see have your cytokines in extracellular space, so the receptors on the plasma membrane, and so on. And your functions and your drugs are outside the cell. So these are sitting outside here. So now that our network is ready and ready to be exported, so you can use these export options that we have. So if you click on this image icon, this is going to let you export the image in any format that your journal requires. You can export the entire image, or you can zoom into a specific section and then you can just save that area. And you do have option for different resolutions. A lot of papers, they ask for 300 and DPI or more as a resolution. So you can easily do that as well. And you can export this as an Excel sheet as well, just to give you the list of all the genes and functions that were utilized as part of this network. All right, so from here, let's move on to our next topic, which is the activity plot. So another great feature you can use within IPA is when you don't, especially when you don't have any data set to upload, is this activity plot feature. So here we can leverage our pre-processed public data sets and identify specific studies where a gene, a function, or a pathway are either activated or inhibited. So here what we have is this activity plot. It looks like your volcano plot, where you have different dots. So here, the different dots represent public data sets where a specific pathway was either activated or inhibited. So a type of question you can ask using activity plot. So I have listed 2 questions here. So here it says, what disease states activate T cell exhaustion signaling pathway? So if you are trying to identify new disease types to see where T cell exhaustion occurs, so you can do that using this feature. You can also find if there are any drugs or treatments that was utilized in the past that led to the inhibition of T cell exhaustion. So this is another way you can query for some of these questions using activity plot. So let's see how we can generate an activity plot within IPA. So for this portion, we are again going to stick to our search box on top. And here this time, what we are going to do is we're going to utilize pathways as an example. So for activity plot, you can use genes, proteins, diseases and functions, and even pathways as a way to query. So here we are going to write T cell exhaustion. So here, while I'm typing in, there's the pathway that shows up. It says T cell exhaustion. So if you are working with some other disease area, and let's say you want to search for neuroinflammation pathway, if you're a cancer researcher, you want to look at tumor microenvironment pathway. So you can search for your disease specific or the research area that you're looking at, search for those pathways here. And this is how the pathway will show up. So here we're going to, again, check the box next to our pathway name. And once we do this, you're going to see this activity plot button that shows up. So we are going to click on this, and this is going to open up the activity plot for us. And we're going to take this step by step to understand how to interpret this activity plot. So by the way, so this is like a very popular feature. A lot of biotech people, they like to use, a lot of pharma companies, they like to utilize this feature for their research. for doing drug discovery and all. Okay, so here what we have is our activity plot. So on the x-axis, we have our z-scores. On the y-axis, we have p-value. So the z-scores are telling you whether your pathway is activated or inhibited. So you remember when we looked at our blue color and orange color, so those would be your activation state or inhibition state for this T-cell exhaustion signaling pathway. So here in this plot, each dot represents one particular one public analysis. So if you go from 0 to right, you can see what is relatively up or turned on based on these positive z-scores. If you go from 0 to left, you can see what is relatively down or off based on these negative z-scores. And the p-value is telling you which public analyses were significant. So this is why as you go up, these public analyses are more significant. So from here, you can ask a question like, so what was our question here? So which disease state activates T-cell exhaustion pathway? So if you want to answer that question, you're going to select the dots on the ones with positive z-scores, because you want to see where T-cell exhaustion was activated. And these are all the public studies where T-cell exhaustion was activated. So you can select these dots by just simply drawing an invisible box and selecting these dots here. And this is going to give you a list of all the public analyses where T-cell exhaustion was activated. So here in the bottom, we have a table with the list of all the public analyses. So these are descriptions of these public analyses. It tells you where these projects are coming from. So whether these are coming from human diseases, oncology related diseases, or links data, etc. So the p-value obviously tells you how significant this was. And the z-score is telling you the higher the z-score, the more activated the pathway was in this particular public data set. This column is giving you the name of the diseases. So here you can see we have breast adenocarcinoma, we have influenza, so a lot of infectious diseases are here, a lot of cancer diseases like small cell lung carcinoma, etc. You can see in some treatment related studies, what were the target genes that was utilized. So here we have HER2, ALK gene, and CDK2. So these are some of the target genes that was utilized in some treatment related conditions. This is telling you the tissue information. So here you can see which tissue was utilized in these different public studies, what drugs were utilized as a treatment. And these two columns tell you what type of comparison was performed. So you can see a lot of these treatment-related studies had increased T-cell exhaustion after the drug treatment. And you also have the link to the source, so where this public data is coming from, some geo page, so you can go to this hyperlink and look at the papers as well. So here our question was, which disease state T-cell exhaustion is activated? So what we can do in that scenario is we can filter for some of these columns. So here we want to look at diseases instead of treatments. So we can click on this funnel icon and say, I want to look at only disease type comparison. So here I'm putting an asterisk after disease keyword. It means it's going to read everything after the word disease. So we'll click apply. So now you can see these are all the diseases where T-cell exhaustion was activated. And here you can see we have Alzheimer's show up. We have influenza. We have lupus, another Alzheimer's, we have leukemia, etc. And then we have ALS as well. So interestingly, just by looking at this, you can see that a lot of neurological diseases are showing up. where T cell exhaustion pathway was activated. So this is a novel interpretation you can get just by not utilizing any of your own data, just leveraging public data sets. And let's say you're interested in this particular study. So you click on this row for Alzheimer's disease, and you can read the information about this paper. So you can see these were, so they utilized a mouse model. So they compared their disease versus control model using, so this was in mouse, and they utilized hippocampus tissue. And if you scroll down, you might even find some more. So these were frozen tissues. And this was processed using NGS. So here you see this is Affymetrix data, and you have the link to the paper. So all this information is already available, and then you can make your own decision whether this is something relevant to your research area or not. So if you are looking for a specific disease type, you can easily, again, filter for a disease. So let's say you just see Alzheimer's. So I can just type in AD, click apply. So now you can see at least 22 public data sets related to Alzheimer's were activated, where T cell exhaustion was activated. So these are like different ways you can utilize public data set. You can leverage public information and find novel insights that you aren't expecting from these type of analyses. So just with a raise of hands, how many of you would find this feature useful for your research, for generating hypothesis, maybe for grant writing? So I see there are some hands being raised. So that's a good sign. So this is an activity plot feature that you can utilize. Similarly, if you wanted to answer the question like which drug treatments predict to inhibit T cell exhaustion, in that scenario, you would basically select all these dots on the left-hand side. So these are all public studies where there was inhibition of T cell exhaustion signaling pathway. So that's another way you can answer some other type of questions. So so from here, I wanted to take a few seconds and to ask if you guys have any questions. So here another question came up. How is the Z-score computed, and what does it speak to on strength of Z-score? So I will, let's see. So let me find that right slide deck to give you an idea how the Z-score works. All right. So let's say you have a pathway that you are looking at. So when we construct any canonical pathways or functions that we utilize, we have some public information that we are already overlaying and telling that this is how a typical pathway should work based on the existing literature out there. Now, when you are overlaying your own expression data on top of this pathway, and say there is a match between the direction of information or the fold change values between your data and this public data, IP ends up calculating a Z-score. And in that scenario, you'll end up getting a positive Z-score, and your pathways would look orange in color, essentially, which is activated. And similarly, if you overlay expression of your data that is working in an opposite fashion of what this original pathway should look like, you will end up getting a pathway which is inhibited, and you will end up getting a negative Z-score, saying that based on the expression pattern that you have in your data set and what is expected from the literature, the pathway was predicted to be inhibited. Now, if we go a bit deeper into how this Z-scores are calculated, Imagine you have a list of genes. So these are your list of genes, and this is the expected direction information that is coming from literature. So we expect that gene 1 should be going down, gene 2 should be going up, and so on. And let's say you have your own list of genes. So these are the same genes, and then they're also behaving in a certain way. So you can see that these two genes are matching in terms of direction. So these were down in the literature, and they're also down in our data set. Similarly, these genes, they were up based on the literature information, and they were also up in your data set. But then if you look at the last pair here, in literature, it says this gene was supposed to be up, but in your data set, this gene expression went down. So what it does is IPA goes and takes into consideration, puts it in this formula. So it's going to take all the matches that we have. So from our list of 8 genes, 7 were matching, right? And then we are going to take the number of mismatches. So here there was at least one mismatch in terms of direction. And then we are doing a square root of the total n. So here you can see 7 minus 1 divided by under square root of 8. So you end up getting a z-score. And if the z-score is positive, then it is predicted to be activated. If the z-score ends up to be a negative value, your pathway was predicted to be inhibited. So this is how we essentially look into the z-scores. So the higher the z-scores, if the z-score is more than 2 and above, it means it is highly significant. If it is -2 or below, it is considered significant as well. So anything in between +2 and -2 are typically not considered significant. But then if depending on how your analysis was run and what pathways showed up, you can then interpret the data in a slightly different manner. But these are the typical numbers that you should be following, 2 and -2, to make sure your pathways are either significant or not. All right. Any other questions related to what we went so far? Don't be shy. Just put in any question. Even if you think it's not relevant, if you think you didn't understand something, just post it there. It's going to help not just you, but all the audiences who might be, the rest of the audience who might be probably wondering about it as well. So I'm going to count in my head. Okay, Mississippi, 1 Mississippi, 2 Mississippi. Okay, so I don't see any questions, but please keep typing in the questions and we will answer everything along the way. So let me go back to our next topic. All right. So after studying, let's say, a signaling pathway that we looked at, for example, T cell activation or like T cell exhaustion, and after doing such analysis, or you find a gene or a target that looks interesting to you, and you want to target this gene for your next CRISPR experiment, or you want to plan an experiment using a drug. So the first question you may be asking yourself is, how is your target gene expressed in different diseases or the disease of your interest? Or is it expressed in the tissue type that you're working with or the cell type that you're interested in? So can we determine how this gene is expressed across maybe known drug treatments? How is this gene correlate with another gene that you might be working in the lab? So this is where Land Explorer feature can help you. So Land Explorer is an extension of IPA, which makes it easy for researchers like you to explore disease-relevant data. So it serves as a central location to vast amount of transcriptomic data, and you can visualize this data in many different ways. So we have basically 3 different types of collections. So when it comes to this omic data, transcriptomics data, we have Oncoland Collection. So if you are a cancer researcher, you can look at some cell line expression data. We have a lot of consortium data coming from sources like TCGA. We have also oncology projects. So these are peer-reviewed papers or clinical trial related data that is stored as part of this Oncoland collection. The next one is our disease land collection. So this is this would be your non-oncology related diseases. So if you're working with infectious diseases, neurological diseases, immunological diseases, so those will be under your disease land collection. So this is divided into your human, mouse, and rat diseases. And this does not only have your expression data, you'll also find mutation data, copy number variant data, etc. And then finally, we have body map collection. So this particular collection will have your normal tissue expression data coming from GTEx, Blueprint, and Human Protein Atlas. So if you're looking for healthy tissue expression, you can simply use some of these collections within the Land Explorer. So before we move on to some of these features, I would like to make a request to all the audience here. So can you type in your disease area that you're working with? If you're working with a specific disease type or a research area, can you put it in the chat box for me, like Alzheimer's disease or like infectious diseases and so on? So that will help me determine what you guys are working with, and maybe we can have a volunteer who can give me their favorite gene or a disease. All right. So here, using the Land Explorer, a simplest question you can ask is, how can we evaluate a target gene or a gene of your interest? So how is your target gene expressed in different diseases or normal tissue types? So what we have in this plot is on the x-axis, we have gene expression. On the y-axis, we have different disease categories. And each dot that you see here is coming from a specific human patient sample, or if it is in a mouse collection, coming from a mouse model. And then you can see how your target gene is expressed across different diseases. Another question we can ask is, how is your gene expressed in specific tissue types that you might be working with? So here you can see that we are looking at ICAM1 gene, and on the x-axis we have gene expression, on the y-axis we have tissue, and you can see that ICAM1 is relatively high expression, has high expression in small intestine compared to maybe something like ileum. So these are the type of distinctions you can easily do coming from public data. Now here, let's say you're working with a specific disease type that you're interested in. You can easily look at the gene expression in a specific disease. So here you can see ICAM1 is expressed in both Crohn's disease and ulcerative colitis. If you were to look for a disease like Alzheimer's or NASH, or so those can be easily possible using some of the filters that are available. Now, a lot of our users, they also like to visualize how two genes might be correlated with each other. So you might be working with some genes in the lab, maybe a list of genes, and you want to see how your target gene is correlated in a specific disease or a tissue. So here you can see that we have a correlation plot for ICAM1 expression and PPAR alpha expression. So here on the x-axis, we have ICAM1, on here we have PPAR alpha. And here we are looking at all inflammatory bowel disease related samples. And you can see there is a negative correlation between ICAM1 and PPAR alpha here. So you can easily plot these correlations using the Land Explorer. In case you're working with a signature, so let's say you have more than one gene and you want to see how the expression looks like in, let's say, in your disease condition or maybe after a drug treatment. So you can generate these heat maps very easily by searching for your multiple genes. And here you can see how PPAR gamma C1b is up in some of these samples, but it went down in some other samples when you sought for a specific disease category or a disease state. And in case you want to find specific projects, so if you're interested in searching for a project related to a drug treatment where somebody was maybe performing a clinical trial, so if you're looking for such information, you can all easily search for them using like a project ID, like a GSI ID, or just simply searching for the disease area. And then you can find these comparisons, the volcano plot for comparisons, where let's say if it was a patient data coming from clinical trial, you can see how complete patients with complete response towards a drug look like versus like a partial response towards a drug. So you can search for these public projects as well, and then you can see which genes were significantly upregulated, and you can export that list as well. So let's see how we can generate some of these views using the Land Explorer. So using the Land Explorer, to use the Land Explorer, we are going to start with the search bar here on top. So so under genes and chemicals, we can search for our favorite gene. So I'll go for a volunteer. Anybody would like to type in a gene that you would like to see in the Land Explorer? You can just type into the chat box, Q&A box. Okay, so I see some volunteers. So let's take a look at one of these genes here. So I see .1L, is that? Yeah, I think it's L. So here, this is a phosphatase. If I search for it, this should show up in the search box. And here you can see this is the gene that we looked at, and you can see this is a phosphatase. And so to open this up to see how this gene is expressed within the Land Explorer, we are going to click on the hyperlink where this is shown in yellow highlight. So we are going to click on this hyperlink, and this is going to open up the Land Explorer. This is going to open up IPA's gene view page. So this is going to open up a page where you can read more about this particular gene, where it is located, if it is involved in specific pathways, etc. So it's going to take a couple of seconds. Oh, so the person says it is not a phosphatase. Interesting. So maybe we can try another gene. Let's see, another gene here. So we have MIC. So we can try this gene. So can the user tell me if the .1L, if it was not phosphatase, what it was? It's interesting that it shows up as phosphatase in our system. I can make a note of it and then see if it was the case or not. So here we search for another gene. We are going to the same thing. So we are going to click on the hyperlink. And this is going to open up the web page for us. Now, sometimes what happens is if you are looking for a specific synonym for a gene, if it shows up differently, like in our case, what happened, it showed up as phosphatase. What I would suggest is to go and type in another synonym, like a known alias for that gene, and that might change the search results. So that is one way to search for the gene that you're looking for. The colloquial name might be different versus the standard gene name might look different. So sometimes that's another way to look at some of these genes. All right. So here we have a MYC, and you can see this is an oncogene, and you can see where it is located and which pathways it's involved in. And if you scroll down, you might find some drug information and some other relevant information. Now, to access the Land Explorer feature, you're going to utilize these links that are available here. So here, if you're looking for normal expression, your gene expression in normal tissue, you're going to utilize these hyperlinks. If you're looking for this in oncology data, you're going to go under these oncology related links. So you have cell line data from different cancer cell lines. We have consortia data coming from TCGA or like Target, etc. And then you have individual peer-reviewed papers that were published. So these would be studies coming from these other links. Now, if you are a researcher who is working in non-oncology data, you are going to go for this disease study. So if you're looking for human disease expression, mouse or rat, you can use those links. So there is a question. It says, what's the backend sources IP collecting all the relevant information? So we do have all this information is coming from various sources. So these include your third-party external databases, like let's say OMEM or like some other public sources. These are all, majority of this information is coming from our curated from manual curation. So we have over 200 curators who regularly go through all published papers. So we read those papers and we curate relevant information coming from those particular papers. So remember when we constructed a network, all these connections had some papers that were cited, how some of these functions were associated. So all this information was coming from those curated papers. And so that is what the wealth of our knowledge base has, which has over 8 million data points. And that is where all of this findings information is coming from. Now, when it comes to the Land Explorer, these are all transcriptomics related studies that were published. So these are coming from sources like GEO or SRA, or for example here, TCGA, Target. So these are all consortia data, a lot of clinical trial data that was published. So these are RNA-seq or microarray related data that has been collected, and they have been processed through our same pipeline that we have using same normalization methods, so that you can easily cross compare the expression data, so that you don't have to process all this microarray or RNA-seq data yourself. This is all has been processed in a similar fashion, in a standardized fashion. So you can do this cross comparison by looking at this huge collection and just searching for your favorite gene and filtering for, let's say, tissue type or disease type, etc. I hope that answered your question when it came to the sources where IP is getting. Okay, thank you so much. So here, since this is an oncogene, I'll first show you some of the oncology collection. And then since we have a lot of public coming from using, like who are working in other disease areas, we'll also go through some examples there as well. So I'll start with this TCGA collection. So we are going to click on TCGA here, and this is going to open up the Land Explorer for us. And this is going to look very similar to the screenshots that I had on my slide. So here, we are going to end up getting this plot where you can see how MYC expression looks like in different in different tumor types. So here on the x-axis, we have gene expression. On the y-axis, we have different tumors. And each circle that you see here in the plot represents one patient sample where MYC was expressed. So if you were to select some of these highly expressed samples, you can just draw a box and select some of these dots. And this is going to open up a table for you. And this is going to give you the expression information coming from the samples that you selected. And you can see that some were coming from skin cancer, some from lung cancer, etc. And then you have some brief information about the copy number or what type of sample it was, if it was a metastatic tumor or primary tumor, etc. So, and you can easily export this information into the Excel sheet and save it on your computer if you wish. So this is what the first search would look like. Now from here, what we can do is we can see how this gene is expressed in different tissue types, for example. So right now, this chart, this plot is showing you the y-axis as tumor type. Now you can easily change this into, let's say, tissue information. So here, if I go under this drop-down menu, we have tissue. So you can select that. And this is going to tell you how MYC is expressed in different tissue types. So you can see it has high expression in the eye and probably relatively low expression in thyroid. Now, if you were to group these samples, instead of tissue information, let's say you want cell type or maybe different tissue, tumor and normal tissue, you can click on tumor or normal. So this is going to separate and group your samples based on tumor and normal. So now you have all your tumor samples in one bar and then all your normal samples in another bar. So using this grouping option, you can keep changing how you group your samples, and it will keep changing your y-axis. So if you want to do gender, you can go back there and see how different genders, where MYC can look, how MYC is expressed in different genders. So let me revert back to our tumor. So I'm going to go back to our tumor. And then this information that we just see has no like real value. If I just club all my tumor samples and say, tell me what it looks like in different genders, then it doesn't make any biological sense. What you can do is you can easily filter for a specific tumor type that you're working with. So here on the left-hand side, we have different options. So if you go under, let's say, tumor type or disease state, you can easily find the relevant tumor that you might be interested in. So under disease state, you can see we have the full names of all these tumors. So we have glioblastoma, like maybe lung adenocarcinoma. So let's say we want to focus on lung cancer. So you can say check none. So we are going to remove these filters and then just select for the ones that we are interested in. So we have lung adenocarcinoma and lung squamous cell carcinoma. And we can apply this filter now. So now this chart is going to limit the search to only lung adenocarcinoma and lung squamous cell carcinoma. Now from here, if you want, you can then go back to the grouping and then say, okay, I want to see all the different samples, tumor types that are there in this cancer type. So here, if you click on sample type, this is going to tell you how this gene was expressed in primary tumor and normal tissue and recurrent tumor samples. So depending on the type of cancer you're selecting, you can also then divide your samples and say how the expression looks like. Now from here, let's say you want to look at cell type information. So you can go back here and say, okay, I want to look for cell type. But if you scroll down and try to read, you don't see a cell type option, right? There is no cell type here. So how can we find cell type information? So to do this, we are going to use this add filter option. So we have a lot of these metadata terms that we call. So sample type is a metadata term, tumor type is a metadata. Now imagine if you want to find some clinically relevant metadata. So let's say somebody did a genetic testing. So you want to group your samples based on this particular genetic testing, or you want to detect samples that had estrogen receptor. You want to look at HER2 positive, HER2 negative samples here. So you can search and find the relevant metadata. You can even find some histology information, a lot of relevant clinical information as well. So you can simply type in. So here I'm going to type in, let's say, cell type. So this might look different. So here if I write cell type, it doesn't, there are a lot of options that show up. So maybe if we go down, we might find the right information. Or maybe if you type, let's say, histology, so you're going to find, like, some histological information. So this is something you can easily search and add as like a metadata term. And this is going to show up here. So here you can see these are the different histological features that was performed in all these different samples. And you can maybe now group your sample based on histology. So you can filter for it. You can also group your samples based on histology as well. So this add filter button is going to allow you to find a lot of relevant metadata terms that you would not find that easily. And you can simply now search for samples, filter for samples based on this information. Now, let's say here we looked at the gene expression. What if you want to look at some mutation information? So if you wish to look at some mutation information, you can go under this view option here. So it says gene FPKM. So right now we are looking at expression values. If you want, you can look at, see, transcription, transcript values. So you can have transcript information, copy number variant information. Here you can look at somatic mutation distribution. So you can click on that option and then it will tell you what type of mutations were observed in your samples. If you want to find more options, you can click on this wheel icon and then you can see we have a lot of other information. We have fusion, then we have a paired fusion information, different types of survival data. So if you want survival data, you can add that too. So I'm going to add all of these here and click OK. So now you have a lot more options available in the drop-down menu. See this, we have now survival data that we didn't have before. So if you want to plot a survival curve, you can easily select survival data, and then this is going to plot a survival curve for you for both your tumor types. So let's see, so these are some of the views that is part of the cancer data that we just looked at. So here we looked at TCGA information. So if you are working with non-oncology data, what you can do is you can go under select land here and then just browse through some of the collections we have. So here we have human disease, mouse and rat disease. So let's say we go under human disease and select that. So this is going to give you the non-oncology data. And this is going to open up this default page where it's going to tell you the number of samples that are available for different disease categories here on the y-axis. And you can easily search for your gene. So here, let's type in another gene from our list. So someone typed FLI1. So we can search for this gene, type in and click search. And this is going to look at the gene expression across different disease categories that are non-oncology. So here we have different diseases. We have like pancreatic disease, we have lots of different diseases, and you can see this is a very busy chart. And maybe we should limit our search to a specific disease category. So now again, just like we did with the cancer data, if you use the options on the left-hand side, you can select for a different disease category. So here, for example, if we look at, let's see, maybe infectious disease. So I'm going to uncheck this and invert my selection. So now we are left with maybe, so let's see, I think we can choose one that you guys mentioned, what disease areas you're working with. So I see some Alzheimer's here. So let's do maybe neurological diseases. So here we have an option for nervous system disease. So I check that and click apply. So now you can see these are the samples that are part of nervous system disease, and this is how our gene is expressed in these samples. Now let's say we want to group our samples based on different disease states that are part of neurological diseases. We are going to go under grouping here and say, I want to group them based on individual disease states that are part of this big category of nervous system disease. So here now you have different neurological diseases. So maybe something like epilepsy has relatively high expression of this particular gene. And then maybe frontotemporal lobular degeneration has relatively low expression when it comes to this particular gene that you're looking at. So this is one way to identify how your target gene is expressed in different diseases. And if you want, you can group this now based on tissue information. So under grouping, we can go under tissue and see how the expression looks like. So definitely peripheral blood had like a very high expression. Interestingly, brain blood vessel also had high expression. So that's an interesting feature. And then skin and spinal cord had relatively like negligible expression. So this is one way you can decide. Let's say if you're thinking of planning an experiment where you want to work on this gene, so maybe this will provide you with some information which tissue type you should be selecting from, let's say, your samples that you're collecting. And similarly, let's say if you want to find correlation between two genes. So here I have one gene and I want to see how this gene is correlated with another gene. So I can type in, let's say, ICAM1, for example. Now you have two genes here and click search. And this is going to give you a correlation between these two genes in nervous system disease samples. So right now, what we ended up with was average expression. Now you want to change this to a view that you are interested in. So we are interested in correlation view. So let's see, we have average. That's interesting. Let's see, we should have more options now. So here we have multi-gene correlation view here. So we can select that. And then this is going to generate a correlation for you, a correlation plot. So this should generate anytime soon. So weird. Oh, there we go. It's running now. So this is our correlation plot between FLI1 and ICAM1. And this is a very busy plot. Now, if you want, you can filter this down to, let's say, Alzheimer's disease. So if I go under disease state, and then we can click on these three dots and let's type in Alzheimer's. And we can select Alzheimer's disease, click OK. So this is going to then filter for samples and will only show you Alzheimer's disease related samples after filtering. So with a raise of hands, how many of you would find Land Explorer useful for your research? All right. I see a lot of hands are being raised, so thank you for doing that. And here, if you want to add some statistics to this correlation plot, you can click on the show p-value, and this is going to show you the correlation. So it has like very low correlation, like of 0.2. So this was not the greatest correlation, but then if you were working with something relevant to your research area, you might find the ones with high correlation as well. And you can easily export any of these images by clicking on these image icons. So any chart, any plot that we generate here, you can simply save it on your computer as well as an image. Now, we do have some advanced options that I have not talked about. So here we have this custom option for both available in oncology and this non-oncology collection. So you can generate custom groupings. So let's say you want to divide your samples based on samples based on for example, FLI1. So you want to see genes that have high expression for FLI1 and want to see which samples have low expression for FLI1. You can also do this in cancer collection. You can do this based on mutation type. So you want to divide your samples, group your samples based on wild type versus mutant for MYC. So you do have these custom options. So if you guys are interested to learn more about this Land Explorer feature, I would suggest to go under our menu option here on the corner and then go to help button. And then this is going to give you different types of views you can generate using the Land Explorer. So here you can see different types of views that you can easily generate, and you can follow the directions and answer lots of different questions utilizing this feature. And if this is something a lot of people are interested, we can always have a specific session on Land Explorer and we can go over some of these things in detail as well. So please reach out to your coordinator if this is something you guys really want to work with. Now, in addition to analyzing your own data within IPA, you can use these public data sets coming within the Land Explorer. So these, like I mentioned before, all these public studies are coming from sources like GEO, SRA, TCGA, or Array Express. And you can perform different types of analyses and you can identify biomarkers, targets, etc., using these public data sets as well. So if you know that if you have tried analyzing public data previously, you would know that it's a long and painful, tedious process. You would have to download this data from SRA. You'll need an SRA toolkit. You'll need to be proficient with command lines. You have to QC the data, generate the counts from the reads, perform secondary analysis, and then you'll have a differential expression data. Then you can upload it into IPA. Imagine how much time and effort will go into this process. And then imagine if you have to do this for hundreds of or thousands of data sets if you are really interested in working. So what our team has done is they have introduced new features in IPA where you can easily leverage these public data sets so that you don't have to go through this hassle of collecting the data from public sources and process all these steps. you can easily search for these public data sets and perform pathway analysis on them. So let's say you read this paper online. You found this on PubMed, and you're interested in this paper, and you want to see how, and this was specifically, let's say, an RNA-seq data, and you want to look at which pathways were enriched in this particular analysis, and without downloading and processing this data. So in IPA, it's as easy as searching for like the PubMed ID. So if you have a PubMed ID from a paper that had some microarray or RNA-seq data, you can easily search for it within IPA, and you're going to find all the comparisons that were performed in this public analysis. So here you can see they were comparing the right hemisphere, which is the Parkinson's disease predominant region versus control. And this was an NGS data coming from human samples. So you can easily search using either PubMed ID, you can search using keywords, and then find the relevant data and perform pathway analysis. So there is a question that came up in chat box. Could IPA load and overlay multiple data sets from different studies? Yes. So you can easily either take your own data and then overlay multiple data sets on top of a pathway. You can take any of these public data sets that I'm showing you right now and then perform similar analysis. And I'll show you how to generate some of this, how to utilize IPA for multiple data sets. So once you find a particular comparison that you're interested in from this public study, you can easily then look at canonical pathways that are enriched. You can easily identify which pathways were activated, how they were involved in this particular disease area. You can open up a pathway and see how the particular pathway was behaving based on the gene expression pattern that was observed in that public data set. You can also look at some diseases and functional ontologies to see how some of these functions behave in this public data set. So you can see that immune cell trafficking, cellular movement, they're all highly activated in this Parkinson's disease data set. So let's see how we can search for public data sets in IPA. So we looked at the Land Explorer by using the search bar, by searching for a gene, and then you wanted to see how a gene was expressed across different studies. Now, using this project manager window available on the left-hand side, we are going to use this search button to search for public datasets that have been pre-processed and stored within IPA. So here, let's type in, for example, I know some people are interested in Alzheimer's, so I can write in Alzheimer's disease and click search. And this is going to find all the Alzheimer's disease related data sets that we have within IPA. So all these pre-processed RNA-seq or microarray data is coming from public sources like GEO, SRA, TCGA, etc. So here you can see we have we have lots of public data sets coming from mouse, human, and single cell data. And here you have the list of those public studies, right? So these columns are telling you what were those studies. So if you click on one of these rows, so this is going to show you what that particular public study looked like. So here somebody was comparing disease versus normal using a mouse model. And this was the genotype probably that was utilized to generate this mouse model. And if you scroll down, you're going to even find, so this was a microarray study and link to the GEO page as well. Similarly, you can go down the list and find another study where they're probably performing some other type of comparison. You can see what somebody was comparing different cell types here, microglial cells. So different types of studies are available, and you can easily open up any of these analyses, or you can compare them as well. So the question was, how can I compare multiple data sets? So you can maybe select some of these public data sets that you like. So I selected for 5 studies here, and then you say add to comparison, and then you can click on view comparison, and this is going to generate a heat map showing the comparison of all these public data sets together. And we are not going to go over that feature in detail right now, but I'll show this feature later on in the last the last part of the session. But I just wanted to still point out, you can compare public data sets even without you having your own data at all. So this is going to generate a heat map for us. So here you can see these are our public data sets, and these were the pathways that were enriched in these public data sets. And you can see how these pathways are behaving within these public analyses. And we'll go over this topic a little bit later. Now, we were specifically interested in that particular paper. So we have our PubMed ID here, right? So you can simply take that PubMed ID, go to the search box, then type in the PubMed ID, and then click search. So this is an easier way probably to search for a project. Like if you search using a broad term, you'll find like tons of information. If you have a particular paper in mind, you can then search using a PubMed ID or GSI ID that usually these studies have, and then you can search for it. So this is going to search for this paper. So these are there were at least 9 different comparisons that were performed in this particular study. And you can see that they compared different tissue types. They had different types of comparisons available. So if you click on one of these comparisons, you can see, so this was right hemisphere, and they did Parkinson's disease symptom non-dominant region versus disease control. And they took a prefrontal cortex from human patients. So similarly, you can click on this other one. So it says right hemisphere, Parkinson's disease symptom dominant region versus disease control. So let's say you're interested in this particular comparison, and you want to look at the pathway analysis for this particular comparison in this paper. So you can double click on this row. So you saw, you highlight that row once, you can double click on it, and this is going to open up the analysis for you. So it's taking a little bit of time to open up. There we go. So now you have a public data set that you did not upload the data, you did not process this data. You simply searched for this in IPA, and then you double clicked on the analysis, and now you have all the results coming from that analysis. So here in the first tab, you can see we have a summary tab. It tells you what top pathways that were enriched in this data set, what were the top regulators, top diseases and functions that were enriched in our data set. If you click on the graphical summary, this is going to give you brief information about how the major biological events that were part of this analysis are constructed together, how they are linked together. So it gives you a big picture overview for this particular analysis. So once this opens up, we'll go over the interpretation of this particular network. So just to revise the steps, what we did so far, we went to our search bar in the project manager window, and then we just typed in the PubMed ID. You can type in your keywords like breast cancer or Alzheimer's or any other disease that you're working with. And then you can simply double click on the particular comparison that you're interested in. And this is going to open up the analysis for you. I have seen like a lot of users who have utilized these public information to basically reinterpret the data in a different fashion. So some people take a list of public data sets and then they compare them side by side and then generate new hypothesis, new biological interpretation. It's like a great way to publish a paper without doing any experiment in the lab or without like spending a single cent doing experiments. So imagine the power of utilizing public data, especially during this COVID time when we have limited access to labs and the access to things. So this is a great way to publish more papers. So here within the graphical summary, this is going to give you a quick overview of the major biological events in this particular analysis. So to construct the graphical summary, our team has utilized machine learning methods to prioritize which nodes to include and how to and to infer the connections between each and every node. So for example, here you can see that this neuroinflammation signaling pathway is activated when IRF7. When this pathway is activated, it leads to increased IRF7 activation, and it also leads to increased IFNG activation as well. So again, orange is predicted activation, blue is predicted inhibition, and this graphical network is going to give you a combination of features. So you'll find pathways that are enriched, right? So these are pathway symbols. We have dendritic cell maturation. So this is another pathway that was enriched. The genes, they're working as regulators. So these are activated or inhibited regulators. And then you also have these functions. So here, these are some functions like stimulation of leukocytes or stimulation of T lymphocytes. So these are functions that were activated as well. So this is giving you just a big picture overview. And the connections also have a meaning. So whenever you see these broken lines, it means there is an indirect connection. So this might be expression-based relationship, or maybe some other players are acting in between. Whenever you see a solid line, so here, for example, a solid line between IL-6 and STAT1. So it means that there is a direct connection. So these could be protein-protein interaction, protein-DNA binding, or maybe some causation relationship. And these dotted lines that you see between two nodes, these are connections and predictions generated by our machine learning algorithm. So this information does not exist in literature. This is something that was generated by the algorithm. Now from here, let's look at the Canonical Pathways tab. So Canonical Pathways feature is probably the most popular feature that our users like to use. And if so here you can look at the key pathways that are enriched in a particular data set. So here on the x-axis, we have the list of pathways that are enriched. On the y-axis, we have the p-value. So the taller the bars, the more enriched these pathways are in your data set. And you'll notice we have different colors on them. So if you see orange color, it means these pathways were activated. If you see blue color, it means these pathways were inhibited in your data set. When you see white colored bars, it means that based on the pattern of expression that we had in our data set, we don't have a clear signal for activation or inhibition. And whenever we see gray colored bars, it means that the z-scores could not be calculated. Maybe we do not have enough published information coming from literature to tell you whether the pathway was activated or not. Now, let's say you are interested in a specific pathway over here. So for example, we are interested in this phagosome formation, which is enriched in Parkinson's disease. You can simply click on the colored bar, and this is going to tell you how many genes from this data set were part of this particular pathway. So 50 genes from our data set, this public data set, were made to the phagosome formation pathway. Out of the 637 genes that actually makes up this pathway, out of which 50 were coming from our data set. And here you can see we have z-scores. So when you have a positive z-score of 2 and above, it means it is significant. The prediction was significant. And this is the p-value of overlap, how significant the pathway was. And just like I explained you guys earlier how z-scores are calculated. So these are the 50 genes that were part of our pathway coming from our data set because they have expression values associated with it. And it also tells you what was the expected direction coming from the literature. And from here, basically, IPA is going to see what was the match or a mismatch between the directions, right? Between your data and this public data, and then you end up with this z-score. So here, let's open up this pathway. So you can click on this open pathway button, and this is going to open up the phagosome formation pathway for us. So here, this is the pathway diagram. And you'll notice we have different colors over here. So whenever we see orange and blue color, these are predictions made by IPA. Whenever you see red and green color, so here we have shade of red and green. So these are gene expression information coming from our data set. Green is downregulated genes that had negative fold change values. Red are the genes with positive fold change values. And so this question that came from Dr. Zhang, who said, could IP load and overlay multiple data sets, right? So now this question arises. Now I have this pathway with me. Can I look at the expression information for this pathway? What was the fold change values look like? And then can we overlay multiple public studies on top of this pathway to see how the pathway would behave in different, let's say, treatments or in other diseases, for example? So to answer that question, we are going to go under this overlay button. And under overlay, we are going to utilize this feature called analyses, datasets, and lists. So here you can see that on the left-hand side, we have a table that lists our data set, right? So this was what we were looking at earlier. And it also gives you the list of genes that made to this pathway. So those 50 genes that we looked at, so here it tells you there were at least 50 genes that showed up, and these were those genes. And it also gives you the expression values for those genes. And if you want to display this information on top of the pathway, you can simply take this option, check the box next to it, and now you'll find expression information. You see this PLD2 has an expression value, and you can see these other genes that were expressed in your data set. So you can have this expression value, and let's say when you're sharing this image with a colleague, then they'll know that this gene was inhibited in your data set, or this gene was upregulated in your data set and whatnot. So these genes were highly expressed. Now, if you want to overlay and compare the expression coming from this particular comparison with maybe another public data set or your own data set, we can go under this Add More button. And here you can browse through your own projects. So let's say you have, some project that you're working with, you can just add it. So I have maybe something like Alzheimer's disease here. So you can select that, click OK, and you can overlay expression data coming from this Alzheimer's data set. And now you can see that at least 84 genes from this Alzheimer's data set made to this Phagosome formation pathway. And you'll see the predictions almost look the same. So if I switch between my two data sets, you see this, the colors change. So this was our original Parkinson's disease, and you can see some of these pathway, the colors changed. And then if I toggle to my Alzheimer's data set, you see this particular section is activated in Alzheimer's, but then this was inhibited in this Parkinson's disease. Now, if you were to look for a public data set, you can go back to Add More and then use this search bar and then type in, let's say, Parkinson's disease and say drug, maybe treatment. So I want to find Parkinson's related data set that had some sort of drug treatment, because I already have Parkinson's data set here overlaying, and I want to see after drug treatment what happens to this pathway. So you can search using keywords, say reset selection and start search. And this is going to find relevant data sets related to drug treatment. So these are all Parkinson's related studies. So here, for example, somebody did a treatment versus control comparison, and this was the drug that was utilized. So let's say you want to use this data set and say add to overlay, and then overlay now. And this is going to overlay this particular treatment related study in Parkinson's human samples. And you can see how different the pathway would look like after the drug treatment. So here you can see there is decreased polymerization of actin, decreased branching of actin filaments and actin remodeling. Versus if I go back to my original disease data set, this completely changes and you can see activation of these actin filaments. So you can toggle in between and you can see the differences. Now, if you want to just simply look at how the expression data looks like in this particular public study, what you can do is you can, for example, click on these charts. So here you see these charts that show up next to the genes. It tells you how the expression looked like in your first data set, which was our Parkinson's disease. Second was the Alzheimer's disease. And third one was drug treatment related study. And you can see how the gene expression changes in these three different studies here. And if you want to, and you can see that there are no values associated with this third study, and that's because the data set was filtered based on p-values or to remove some low expressing genes, but you can ignore those filters if you want to by simply clicking on ignore analysis cutoffs. And you see this gene expression went down after drug treatment. So this is a very interesting scenario, right? So you are comparing a disease condition where this gene went up, but after the drug treatment, this particular gene expression went down. So this is a novel information that you found from just by utilizing public data sets. And you can use this information to maybe publish this, or you can simply write a new paper talking about this unique information that you found. So similarly, you have the same feature here. So you have expression is up under disease condition, and it went down after the drug treatment. And you have a reverse scenario here where LBP was down in disease condition and it went up after the drug treatment. So just with a raise of hands, how many of you would find this feature useful for your research? I see some hands are being raised. I just wanted to make sure we keep this interactive. I know this is a very long session and you might feel distracted in between. So that's why if you guys have any questions along the way, I would highly encourage you to put your questions, and then we can make this more interactive. All right. So we are moving to our last hour, and we'll look into how to basically process your own data. So far, we looked at how to leverage IPA when you don't have any data and you want to just look at public data information and just utilize it for your research. Now we have a scenario where we want to publish our own data. We want to analyze our own data. So let's take a look at this particular scenario. Now, to run an analysis in IPA, you basically need a list. Now, this list can be a list of genes, proteins, metabolites, and this list can be uploaded in a form of a text format or Excel format or diff formats. We do accept a lot of a variety of species that you might be working with. So if you have a gene list using IDs coming from these species, you can easily utilize IPA in that scenario. If your species is not listed here, please type into the chat box and let me know what species you're working with and if it's not here. Now, most typically people utilize their microarray or sequencing data. I have seen a lot of people utilizing, very uniquely, very creatively ChIP-seq data or their CRISPR data or some qPCR or Western blot data as well. So depending on what type of information you have, you can still utilize IPA for your research. Now, if you do have some kind of expression data, you can upload it into IPA. So here we have a data format that you would follow for uploading your data. So here you need to make sure that the first column is always the ID column. So you have some sort of gene symbol, ensemble ID, basically an acceptable ID format. And then followed by fold change values, P values, adjusted. If you have FDR values, you can add them as well. So these fold change values are basically coming from some sort of comparison. So these are comparisons either you're doing between a disease versus normal, wild type versus mutant mouse, or treatment versus baseline, time point 1 versus time point 2. So those are the comparisons you're performing, and which helps you generate the fold change values. So this is basically the format, and it's very simple. Either upload it as a text or an Excel sheet, and you're good to go. Now, in case you're working with multiple experimental conditions like multiple time points, multiple treatments, or different mouse models, you can upload that data together in one big Excel spreadsheet. So following the same format, you have your gene IDs in the first column. The next 2 or 3 columns are dedicated to your first comparison. So this would be maybe your disease versus normal. So we call this observation 1. And then followed by full change values, p-values coming from another experiment, like maybe treatment versus baseline, or let's say time point 1 versus time point 2. So that would be your second observation that you'll have. A third one and fourth one, so you can have basically one big Excel sheet, and then you can have all of your experimental conditions together. So there is a question that says p-values and Fdrs are optional. How do they affect the analysis? That's a great question. And so the p-values and Fdrs are optional. So typically you use p-values when you're working with, let's say, multiple samples, right? Like if you had multiple replicates, you were able to generate a p-value. It comes into the picture when you're uploading the data. You want to filter out your non-significant genes, right? So you obviously don't want genes of low significance into your analysis. So that's where your Fdr or p-values come into picture. You can just type in a filter and say, okay, I want to only include genes that meet 0.05 p-value criteria. So that is where your p-values and Fdrs will come into picture. If you don't have them, you need not worry. You can still work with your fold change values, and you can just define what would be your fold change cutoff. So you want like at least +2 or -2 fold changes as your cutoff. So you can have that as a cutoff as well. I hope that answered the question. So another question came up, is there a maximum number of genes we can input for IPA? I believe the number is 200,000. But when you are analyzing your data, you need to make sure your working list after filtering for p-values and let's say fold change values, you want the most significantly differentially expressed genes to be in your list. And that number is somewhere between, I would say, a low of 100 genes to a maximum of 8,000 genes. And the reason I say that is because imagine if you put all your genes for your analysis, everything will show up as activated and inhibited. You're not going to be able to find the most significantly enriched pathways. You won't be able to find the real biological interpretation from your data if you include like all your 20,000, 30,000 genes. So that is why we limit these numbers to a workable number, which makes sense when generating the results from the data. Now, in case, so just with a raise of hands, how many of you are working with metabolomics by chance or planning to work on metabolomics later in future? I see there are at least 2 people who are working with metabolomics. That's great, actually. So in case you're working with metabolomics data, the format is almost the same with few exceptions. So you do start with your ID columns. So in this case, you have your metabolite IDs instead of your gene IDs. And if you do have, like, let's say PubChem ID, you include that. My recommendation is to include as many ID types as possible when it comes to metabolites, because sometimes when, let's say, what the way IPA looks at these IDs is it's going to first go through the first column. And if it doesn't find or recognize a specific ID, it's going to go and look through a second column and say, okay, so I do find a KEGG ID, and then it's going to include that metabolite. And then similarly, it's going to the third column and fourth column. So these are the 4 acceptable ID types for metabolite data. In case your core facility ends up giving you just the metabolite names, you can use these conversion tools. So ID conversion tools, you can just take your list of metabolites and basically upload them into these publicly available websites, and then it will convert it into these specific ID formats. And then you can basically arrange this Excel sheet with your metabolite IDs, followed by fold change, p-values, Fdr values in case you have it. And on an average, when people are working with the metabolite data, they end up with very small list, maybe 70 or 100 or 200 metabolites. So it is okay. And you can start with a small number and you can still run an analysis using the metabolite data. So acceptable ID formats, and make sure you have as many columns as possible for your metabolite IDs. All right, so today we are going to look at a particular example data set. So this study came from multiple sclerosis patients, where they looked at samples cytotoxic T cells coming from multiple sclerosis and control samples. And then they performed hematopoietic stem cell transplant, and they wanted to see how the cytotoxic T cells look like post-transplant and pre-transplant. So we have two different comparisons. So we have disease versus normal. And the second is your treatment versus baseline. So this is technically a transplantation. I would assume it to be like a treatment versus baseline. So these are our two comparisons. And we are going to basically look at how to upload this data and how to interpret that data. So just with a raise of hands, how many of you have previously uploaded data within IPA? So I see at least like a quarter of you guys have uploaded data into IPA. So I'm assuming that the rest of the crowd has not ever uploaded data. So maybe it's a good time to upload the data and look at the process. So here under create new button, this is going to allow you to upload the data. And let me just open up the Excel sheet, how it looks like, so that you get an idea. So here I have one data set for my MS versus normal samples. So we have gene ID, we have full change values, raw P values, and FDR values. So this is how the Excel sheet is. And we have at least like 15,000 genes here. Okay. So we are going to start with create new button and click on core analysis. And once we do this, we are going to click on this upload button. Now this is going to take you to your computer and you can browse and find the correct Excel sheet, and you can just simply select it, click open. Now, once you do this, you're going to find this useful workflow page. So if you forget the steps along the way, you can keep this open and you can just simply follow the instructions and you'll be able to upload the data. So here the next step is we are going to leave all these options as default. You don't need to change anything. Just make sure if you have a column header, which in our case we did have, we are going to select yes. The rest of it you can ignore and it's not going to matter. Now the second part of the section is this raw data. So here you can see that in parentheses it says 15,822. So these were the rows. These many rows were identified in our Excel sheet. And here we are going to call our first column as ID. ID column is always the first column. So in the drop-down menu, we select ID. And here you can see IP automatically identified ensemble as the ID type. And here we do have lots of other IDs. If you end up using like a gene symbol or like, some Illumina ID, you can use IP will automatically identify this. If you're working with proteomics data, you see we have International Protein Index, we have Uniport ID, so a lot of different IDs are available. And for metabolomics, we have this CAS registry, then we have PubChem ID, etc. And if you are working with microRNA data, we do have microRNA ID types as well. Now, the next step is to call the first three columns as observation 1. Because remember, the observations are nothing but our experimental condition. And all these three values, our log ratio, p-value, FDR value, are part of one particular observation. So this was our multiple sclerosis versus normal. So automatically IP identified this as log ratio, p-value. And this one, you can go down and drop down menu and change this to FDR because this is supposed to be FDR value. So we can just simply change that. Now, if you want, you can change the names of these observation. Instead of calling this observation 1, you can change this and say, okay, I want to call this MS versus normal, so that you can easily remember what experimental condition was, and click OK. So the names are automatically changed. Now from here, we are going to simply save this because we are done. So we have our ID type, we have our values, and they were all here. And then we are going to simply save this. So here it says we do not have any metadata. So in IPA, you do have an option to add metadata information. So let's say you want to say I'm working with cytotoxic T cells. You can just write into experimental information, like these are cytotoxic T cells coming from human species, maybe collected at a time point. So you can add that project level information just to track your projects. And click OK. So here I'm going to create a new folder and I'm going to call it test2, save. And I'm going to leave this. So this is going to take whatever Excel sheet name you had, and you can save this like that. So we're going to save this. So I see there is a question in the chat box about setting the threshold for log ratios and p-values. So I will address this question just in like next 2 or 3 minutes when we reach to that section, and it will be very easy to understand how these analysis cutoffs are set up. Just like bear for like few minutes and we'll get there. So here, the next step is we want to run the analysis. So today we are going to run expression analysis. So in case you're working with some drug-related data or toxicology data, you can run tox analysis. If you are working with metabolomics, make sure you select metabolomics in that scenario. So for your proteomics, single cell RNA sequencing, microarray, all those types of analysis will go under your expression analysis. Now here IPA is asking you what measurement type would you like to use as a basis for this analysis? So it's always going to be a log ratio value, a fold change value, some sort of ratios, right? So you need direction in your data. You need that positive fold change values and negative fold change values, because that will allow you to generate those z-scores, and that will display the colors on your pathways and networks that we see in IPM. So make sure you select for your fold change or log ratio values. And then we click next. So now we are at the page where we want to set up the analysis, where we want to set those cutoffs that we were talking about earlier. So this section can be divided into 2 parts. This first portion will allow you to basically set up different filters if you wish to. So right now, this is in our default settings. If you're running the analysis for the first time, I would suggest to go under networks option. And if you don't have this option checked, make sure this is checked and then run your analysis. So this will allow you to perform some advanced analytics. If you leave it unchecked, it won't perform the analysis. Now, when you're running for the first time, I would recommend to leave these settings as default. And depending on what type of data you're working with, what type of analysis results you are expecting, you can always go back and change these things. Now, when it comes to the fold change values and p-values, this is the section where we are going to define our cutoffs. So here, for example, we have raw p-value. And you can see the range here. And what we are going to do is we can, let's say, type in 0.05 and say, okay, I want to limit my list of genes, which was 15,762, out of which 6,000 were up, 9,000, sorry, 6,000 were down, 9,000 were up. And you want to change this to a workable number by removing anything that is not not significant. So we have this cutoff and click recalculate. And now you see this, we are now left with 4,858 genes that meets this criteria of 0.05. Now this is still, this is a good number to work with. You don't need to worry about it, but then you can make it even more stringent by using cutoffs for this full change value here. So let's say we type in 0.8 as my like a limit of upregulated genes, and then maybe minus 0.8 as a cutoff for my downregulated genes, and say anything above or below we want to include, anything in between we don't want to include in the analysis. So we can type in that number and hit recalculate, and now we are left with 2,385 genes. So this is how you set up these filters. Now, many times, a lot of our users, they ask, what is the right number to utilize here? Now, so that will be a very data specific. What type of analysis you're running, what type of data you have, what type of experiments were performed. You want to use p-values, at least when it comes to p-values, you want to use the industry standards. So imagine when you run this analysis, when you're publishing a paper and you send it to reviewers, right? So when you are sending it to reviewers, you are going to report that these were the p-values we used as a cutoff, and these were the fold change values we used as cutoff. So it's up to you how you're convincing your reviewers. So 0.05 is probably like the most acceptable one. But after you use this, you're not left with any genes or very few genes, then you might have to rethink how you utilize these filters. Maybe you can then say, I want to maybe limit my cutoff to 0.01. And then it's up to you how you are going to convince the reviewers about the cutoffs that was utilized. So it is very subjective depending on the type of data you're analyzing, what type of samples you have, and how you, while writing the paper and interpreting the data, how you report some of this information. But at least when it comes to this particular data set, this is a good number to work with. So 2,300 is a good number to run the analysis with. And from here, we are simply going to click on Run Analysis. So, okay, there is a question about what about Q values? So yeah, so you can utilize your Q values. I did not utilize it in this case, but then you can always use that filter. Like I just kept my p-value as my cutoff, but then you can always type another number for your Q values and see this is going to even remove a string. This is Q values already adjusted p-values, so this is going to be even more stringent. So maybe you can type in another p-value and see if that's going to change your analysis. So this is how you work with some of the filters. You decide which ones make more sense in your case. And another recommendation I would give at this point is try to run your analysis at least 3 different ways. So one with very relaxed filters, one with very strict filters based on your p-values and fold change values, and then something in between. So when you run it in 3 different ways, what will happen is when you look at the analysis results, some pathways, if they keep showing up, even with your relaxed filters and strict filters, you can feel confident about those results that, okay, so this pathway always shows up whether my filters are strict or relaxed. So you can rely on that result much more. And when you're writing up the paper, you can just take that information. So run this analysis 3 times, see if it makes sense like to you when it comes to your biological interpretation for that experiment that you're running. So here, the final step is to click on Run Analysis. And this is going to save the analysis in that folder that we just created. And we click OK. And this is going to show you this clock icon that you submitted your analysis. If the analysis is running, you'll see this wheel icon. And once it is run, your analysis will be ready. So here you can see this has a clock icon. So this was just submitted. It hasn't started running yet. And once the analysis is run, it should look something like this with this expression E symbol. So you know that your analysis is done. And you'll also get like a big a yellow strip at the very bottom of IPAs, which tells you your analysis is ready. And you'll also receive an email saying that your analysis is done. Now, once your analysis is done, your results, so I already ran this earlier. You can simply double click on the analysis. And this is going to open up the analysis page for us. Now, if you guys remember, this is exactly how when we opened up one of our public analyses, you can see the same features here, right? So we have summary page, graphical summary, canonical pathways, etc. So the sections would look exactly the same, but the results will be different for the different types of analysis you're running. So here we know how to interpret the summary page. We also know how to interpret the graphical summary. And here we have canonical pathways. So we obviously know how to interpret some of the canonical pathways here. So on the x-axis, we have pathways that are enriched. On the y-axis, we have the p-value of their enrichment. Now, what I did not show you earlier is some of the other features that are available for canonical pathway. So here, if you click on this customize chart option, this is going to give you a list of pathways that are part of our database. So let's say this is a neurological disease that we are working with. We really don't care about maybe the cancer related data set pathways that show up. So if I, for example, look at under signaling pathways, I have apoptosis, and then we have like these cancer related pathways here. So let's say we don't want this cancer, we can uncheck this, right? And it's going to remove all the cancer pathways and leave the rest of them. And you can click apply. And then this is going to limit our filter and will only limit the pathways that are related to non-cancer. Similarly, you can go back here under the customize chart and say, I want to maybe only limit pathways with at least minimum z-score of 2. I don't want anything that's showing up like below, like, which does not have a significant z-score. So you can write a number and then click apply. So now you're left with fewer pathways, but then with strong predictions. So this is something maybe you can go back and report as part of your results. Now, let's say you're interested in finding pathways which have specific set of genes that you're working in the lab. You know, this is a key gene that I'm working in my area, and I want to make sure that this gene was part of this, some of these canonical pathways here. So you can easily filter by just typing in the gene symbol, let's say IL-2, and then click apply. So if it isn't there, this is going to not report anything, but then, if this gene was present in some of the pathways, it's going to include that and just going to filter that list for you. So these are some of the ways you can change the information. A lot of our users, when they're publishing papers, they also want to change the font size, like because it's small. So you can change the font size for your axes or the labels, and then make sure that when you're publishing, they are in the correct font size that you're looking for. So these are some of the filters that are available as part of canonical pathways. You can export these images, right? Or you can export it as an Excel sheet, and maybe you can replot this in some other visual, like, statistical software, maybe Prism or Excel sheet if you want to, have a standard way of publishing data. You can also maybe, for example, open like one of the pathways. And let's say you're looking at neuroinflammation pathway, and in the supplementary section, you want to report which genes were part of this pathway. So you can export this as an Excel sheet, and then you can add it as a supplementary information within your paper. So these are some of the key features that will help you to, let's say, publish a paper when it comes to some of these data. And I know we went through the canonical pathways and how to utilize the overlay tools and etc. What we haven't talked about is this upstream analysis feature here. So if you click on this section, this is going to give you the results for upstream analysis. Now, a lot of researchers, whether in academic institutions like yours and like, in biotech, they are interested in identifying which genes are acting as upstream regulators and influencing the expression of genes in your data set. So the way we define upstream regulators is, give me a second, and I wanted to show an image to explain how upstream regulators should look like. Unfortunately, I don't have that slide with me, but I can draw and explain what an upstream regulator is. So imagine you have an upstream regulator sitting on top. So this is your upstream regulator. And this regulator is influencing the expression of genes in your data set. So these genes are sitting downstream, and these are your target genes in your data set. So target 1, 2, 3, 4, etc. So this regulator is influencing the expression of your genes in your data set, and that's how it is influencing a function or a pathway downstream. So this is how we define an upstream regulator. So here what we have is the list of upstream regulators that were identified in your data set. And then these upstream regulators can be genes, they can be proteins, and even drugs and chemicals. Because imagine if you're introducing a drug in a system, in a mouse model or in cell line, it is still changing the expression of genes in that cell or in that tissue or in this mouse. So we always think of upstream regulators that are changing the expression of genes in your data set. Next, we have expression log ratio values. So obviously the genes that were identified as part of your data set that are acting as regulators, these are the ones that are coming from your data set. Some of these guys that we have here without any expression values. So these were identified from our knowledge base or our repository or collection that we have, because somehow these regulators were still influencing the genes in your data set. So these are novel ones that were not part of your data set. Now you can focus on a type of regulator that you're interested in. So here, if you go under this filter icon, you can limit your search to only genes and proteins that are acting as regulators, or you can find drugs or chemicals that are acting as regulators, or you can choose from this list. Let's say you are only interested in cytokines. So you can check that box, or you want to only focus on growth factor or kinases, or maybe let's say a transmembrane receptor. So here, let's say if I select transmembrane receptor, click apply, this is going to filter for only for the regulators that are only transmembrane receptors. Now from here, we have the next two columns that tells you if these regulators were predicted to be activated or not. So if you have a positive z-score, these are predicted to be activated. If you have negative z-scores of 2 and -2 or below, these are shown in blue color and they are inhibited. So you can easily, let's say, sort for this. Let's say I only want z-score with at least 2 minimum cutoff. You can just type in that and click apply. So it's going to remove all these regulators without significant z-score values. And next one is a p-value of overlap. So this is telling you if how significant this regulator was. So you can again go here and say, I want to type in 0.05 as my cutoff for p-value and click apply. And then this is going to filter this down further for you. So now you have a workable list to work with. So we started with some 1,000 genes that were working as regulators. Now you have like 18 transmembrane receptors that you can work with. And here we have the target molecules in our data sets. So here the way you would interpret this is, for example, CD40. So this regulator was up in our data set and it is activated. It has a very nice z-score. It is significant based on p-value, and it is influencing 36 genes in your data set. Now, let's say you want to look at this particular transmembrane receptor and you want to open up a network for this. So you can click on display as network. So you need to select the row and then click on display as network. And this is going to open up this regulatory network for you, where you can see CD40 is regulating, activating itself, self-regulatory, and it is also acting upon some of these genes in your data set. So the red colors are the ones where your genes were upregulated, they had positive fold change values. The green ones are the genes that had negative fold change value expression. Now from here, we have a nice regulatory network, but then you want to see how this regulatory network is influencing maybe a downstream function that you might be interested in. So this was multiple sclerosis data set, right? So if you want to identify if this network is influencing a function, you can go under build. And under build, you can click on this grow button. And under grow, we are going to use this diseases and function tab. And this is going to give you the list of all top disease and functional ontologies that are being influenced by this particular regulatory network. So this is going to generate a list. So while this is processing, with the raise of hands, how many of you understood the concept of upstream regulators when we went through the diagram and how this whole thing works? Okay, I see some hands are being raised, so that's a good sign, because it is sometimes confusing how upstream regulators work. It's good that you guys are following the explanation. Okay, so here we have a list of functions, and you can see that which genes in your network are influencing these functions. So you see a lot of function of leukocytes, activation of blood cells, quantity of leukocytes, lymphocytes. So a lot of inflammation related functions are showing up very frequently here. Lymphocyte homeostasis, cytokine, chemokine signaling, apoptosis of so a lot of immune related functions are showing up. So maybe those are something relevant for multiple sclerosis. So that is one way you can interpret that. And let's say we want to add a function here. So let's say cell proliferation of T lymphocytes. I want to add this function to my network. So you can select that function and then click on the apply button. And this is going to add this new node. And you can clearly see that when this regulatory network, the CD40 is activated, it is acting upon these whatever number of genes we had, like maybe 30, 40 genes, that 36 genes that we had. It's influencing the expression of these genes, and then it leads to increased cell proliferation of T lymphocytes. So that's the interpretation now you're getting from this regulatory network. And if you want, you can change this into a, let's say, hierarchical clustering. So now you have like CD40 sitting on top, influencing the genes in your data set, and they're ultimately, it leads to increased increased proliferation of T lymphocytes. So this is how some of the things you can easily generate. You can interpret some of this data, which makes more sense biologically. So you can even take this, like you can add another function here. So for example, activation of leukocytes, and then apply, and this is going to add another function here at the bottom. So now we have activation of leukocytes. And I can move this down, and you can see this is also shown in orange color, right? So now you can feel confident that maybe some of these immune cell related functions are getting activated in my multiple sclerosis data set when CD40 was activated, and it's influencing all these genes, and it's happening via these key genes in my data set. So this is the upstream analysis feature. I think we have like some more time, and I want to focus on two other topics that I wanted to cover. But basically, if you want to look at some of this information down in this list of tabs, you can go into each section, and you're going to find this video icon and this question mark button. And this is going to allow you, this is going to give you a brief information about that particular section and like how to interpret some of the data that's there in this section. So each tab will have this video and question mark button. So let's move on to now a different section of this analysis. So remember, we were working with two different data sets, right? So we had disease versus normal, where disease was multiple sclerosis. And then we had a stem cell transplantation that was done. So that's like a treatment that was performed for multiple sclerosis cytotoxic T cells. So how do you compare these two analysis or these two experimental conditions? So you can easily do that by going under create new button and then clicking on comparison analysis. And here you're going to go under your data set that you're working with. So here I have my multiple sclerosis data set. So I already have this normal data set. Let's add the stem cell transplant data set. Now you have two data sets here. And then click on view comparison. So this is going to generate this heat map where you're comparing your two or more analyses side by side. So here, this looks pretty similar, right? So in our core analysis, we had the canonical pathways, upstream analysis, diseases and functions, and so on. So for comparison, you have the same results, but now you're comparing two or more data sets side by side. So the first was our disease data set. The second column is our treatment data set. And you can clearly see some of these pathways, like, for example, Phagosome pathway, Krebs signaling in neurons. You can see that these pathways were activated, shown in orange color, in our disease data set. But after treatment, you can see these same pathways are now inhibited after the drug treatment or the transplantation. So you can see the difference. And let's say when you're publishing your paper, you can simply take this heat map and show which were the key critical pathways that were dramatically changing after the stem cell transplantation. You can also sort this based on different criteria. So right now, this is heat map is sorted based on z-score. So the ones with highest z-score are placed on top of the heat map. Let's say you want to change this to hierarchical clustering. So you can easily cluster your pathways based on their similar functions. So here you can now have a hierarchical clustering. Some similar pathways are working together. And you can clearly see the distinction of activation and inhibition in your two different data sets. Similarly, you have like all these other pathways that are working in similar fashion. You can also, for example, open up a pathway. So for example, if I go back to score, I liked the phagosome formation here. So here, let's click on the blue colored box for the treatment. And this was the same pathway that we looked at earlier. And you can clearly see the different colors. And if I switch back to the orange colored box for this pathway for the normal data set, the same pathway will now look very different from our drug related data set. So, so this is basically one way. Another way to visualize this information is by clicking on gene heat map. So if I click on gene heat map, this is going to give me the list of genes for our phagosome formation in our two different data sets. And it basically tells you which genes were up or which genes were down just by looking at the colors here. So all of these genes were down in treatment condition, but then a lot of these genes were up in disease condition. And you can change this by clustering. Let's say if you want to cluster your genes together, and you can see the pattern of expression, how it changes. So this is another way you can look at the data. But this is only showing you the genes that are part of phagosome formation. Similarly, I closed the comparison analysis, but then similarly, you'll be able to look at not just the pathways, you can look the heat map for upstream analysis or diseases and functions, etc. And it'll all show up in a form of a heat map. So I believe like one user was asking about this, how to compare multiple data sets. So this is how you do it. Go to create new, click on comparison analysis. Now, the last section I wanted to talk about today was analysis match feature. So analysis match feature within IPA is where you can compare your whole transcriptomics data or proteomics data with other public data sets completely. So you're taking your whole data set and then you're comparing it with these public analyses. So this is located at the very end of the tab, which says analysis match. So what analysis match is doing is it's taking into consideration the signatures that are coming from your analysis, and that would be canonical pathways. We looked at that today. The upstream regulator signatures, the diseases and function signatures, and then this is called a causal network. So causal network allows you to identify master regulators that are sitting on top of multiple regulators. So, sorry. So imagine these were your upstream regulators, multiple upstream regulators that are acting on the genes in your data set. You can easily find a master regulator sitting on top of this big network and see how it is influencing the expression of genes in your data set. So the analysis match feature is taking into consideration all these 4 types of signatures. And the results for analysis match basically looks like this. So you'll end up with a table with information coming from those public data sets. So these are all the public data sets that you had some sort of a match or an anti-match with your own data set. So this first section is giving you just the name of the projects, where they're coming from, if they are relevant to a disease or a treatment, which tissue type information, and so on. Now, the second half of the section is giving you the scores, the 4 signatures that we talked about. So CP is your canonical pathways Z-score. UR is upstream regulator, CN is causal network, DE is your diseases and function. So the score basically runs from a positive 100 to a negative 100. So positive 100, like you see here, is a perfect match, right? So your own data set will have a perfect match with your own data, which makes obvious sense. So all your pathways, regulators, networks, they all have exact like 100% match with each other. Similarly, a negative 100 would basically, ideally speaking, you have a perfect data set that has a perfect opposite match to your data set. So that's how you look at these numbers here, the scores. So the higher these numbers are, the higher the match between the biological signatures between your data and public data. The higher the absolute value of these negative matches, the higher is the, I would say, the anti-match between your data and public data. Now, you might be wondering, why do I care about comparing my whole data set together, like, with these public data sets? So a lot of our users, they like to compare their data with public data is because, one, this is a very nice way to validate your own findings. So imagine, let's say you're working with Alzheimer's disease data set. So you are working with a mouse model and you ran a RNA sequencing analysis, and you want to see how well the model is reflected in terms of these biological signatures. So you can easily find public data sets that use maybe other mouse models or the data sets coming from human patients, and you can see how well your biological signatures from your mouse model is matching with these public data sets. So that's one way to validate your own findings. You can feel confident about your own results. Another way would be, let's say you're working with some organoid model. So let's say you made an organoid that you generated, and you want to see if this model looks very similar to mouse or human data. So you can easily do that matching, and you can feel confident that my organoid shows the same biological signatures that a typical mouse model or a human patient sample would have. Another way would be if you want to do drug repurposing, so you can identify novel drugs that you can maybe you can select them based on these public data sets, and then you can run some experiments in the lab. So a lot of biotech companies, they like to utilize this for drug repurposing utility. So let's see how we can look at some of this information. So when you click on the analysis match tab, what you'll end up with is a big table. So you have your name of the analysis, right? So these are your description for your public data sets. This is telling you whether they're coming from human data or cancer data or some mouse data. Then you have the name of the disease. So it just gives you the names of the diseases. This tells you the target gene that was utilized for maybe some sort of treatment. So this was maybe a treatment related data set where a gene was targeted. Then you have tissue information, which tissue type was collected in these studies. If there was a drug treatment that was performed, you can see that information. And then a type of a comparison that was being performed, so like, treatment versus control, disease versus normal, or maybe some other like cell type 1 versus cell type 2. And these are the scores that we talked about, right? So the higher the match of these scores, the better your data set is matching in terms of these biological signatures. And here you can see we have at least 91,000 public analyses showed up when we were comparing our multiple sclerosis data set with all the public data sets that we have, 91,000. Now that's a lot of studies. You want to filter out and find the relevant information depending on what type of question you have. So let's say I want to find a drug that I can treat my multiple sclerosis samples with. I want to find what would be ideal drug that I can utilize. Now, the logic says that when I'm working with multiple sclerosis, under disease conditions, a lot of these pathways will or functions will maybe go up. So these are all public studies where I have a positive match. So the logic says that when I'm treating my disease with a drug, these signatures will work in a complete opposite fashion, right? Like they should behave very different way than what I would see in a disease condition. So what you can do is you can start filtering for relevant information. So first we'll do is we want to only stick to treatment related data sets. So under comparisons, we can type in treatment and click apply. So we remove every public data set that does not involve a treatment. So now we went from 91,000 to this 49,000 public data sets. Now we want to find public data sets that had maybe a very opposite biological signatures, because after drug treatment, you're expecting that disease or to behave in a completely different fashion. So you can sort this and say, I only want to see public studies with negative z-scores. So I can flip my table to reflect all the negative z-score values, right? And then I can find the highest highest negative z-score value. So these are pretty high, like 55%, 61% negative match. And here you can see that some of the treatments that were utilized was IL-2. I see another IL-2 here. Then so some of these drugs are showing up very clearly. Now, these are treatments that were utilized externally. So whenever you have a metadata that says treatment, it means it was an ex vivo treatment. So if you want to find some clinical trial data, you can go under customize table and say, I want to find clinical trial data. So for that, you want to select subject treatment. So that's in vivo related treatment data. So you can add this as another column. So here we have another column for subject treatment, and this is your regular treatment. So now you can see that some of these cancer related drugs show up. So PD1 was anti-PD1 antibody was like one that keeps showing up where, my signatures change completely. If you don't want cancer related data sets, you can go here and say, I don't want cancer, I only want non-cancer data. So you can choose from these public analysis. So remember when we worked with Land Explorer, we had OncoLand, disease land collection. So we select disease land collection and click apply. And now you're left with non-oncology diseases, and then you can maybe look at some of the treatment data that you end up with. So see, these are some of the top hits. And if you want, maybe, let's say if you want only neurological data, you can look and add maybe therapeutic area as an option. So you can select a column and add therapeutic area. And that's another column that you can add that you can use to filter for. So here we have therapeutic area. So and say I only want neuro and click apply. You can just type in neuro and then you can click apply, and then you find all neurological diseases. So these are some of the drugs that were utilized for neurological diseases where the pathways and the biological signatures were working in a completely opposite fashion. So maybe some of these drugs can be repurposed for multiple sclerosis. So with the raise of hands, how many of you would find this useful for your research? I know this is the end of the session and everyone is tired. But yeah, I still see some hands being raised. Whew, that's good. I think you guys have still some energy left in you guys. So thank you for raising hands. And I see some of you would find this useful. So this is how you use the analysis match feature. And let's say you found these studies interesting. So you can select some of these studies and say, I want to view them as a heat map. So you can basically compare these